Protein synthesis involves transcription and translation. Transcription creates single-stranded mRNA that are instructions for the foundation of proteins. This happens inside of the nucleus, and an enzyme helicase breaks hydrogen bonds between the nitrogenous bases so RNA polymerase, going from 5' to 3', can lay down complementary RNA strands to the template encoding strands. The mRNA then leaves the nucleus through nuclear membrane pores. Translation happens inside the cytoplasm and on a ribosome that is made of ribosomal RNA. Meanwhile, tRNA inside of the cytoplasm carries amino acids and eventually has the function of bringing different amino acids together, but it needs mRNA's instructions for how to do so. mRNA tells which tRNAs to come to the ribosome and pair up with mRNA bases. The complementary bases are read in triplets, or codons. tRNA will have anticodons of uracil, adenine, guanine, cytosine that will pair with the codons of mRNA, which also have the nitrogenous bases of uracil, adenine, guanine, cytosine. Each time the tRNA's anticodons will pair with the mRNA's codons, an amino acid is transferred. As more amino acids are transferred, they are held together by peptide bonds until a stop codon appears, which is the indicator of the end of transcription or translation. The result is a polypeptide, which is a polymer of protein macromolecule, and is used as a variety of functions throughout our body.
DNA replication is when DNA is copied and then replicated to be used in another cell. This is done beginning with helicase unwinding the DNA for it to be remade. When the DNA is split, primase comes and lays down primers on the old DNA strands from 5' to 3', with the top strand of DNA being the leading strand, which is 5' to 3', and the bottom being the lagging strand, 3' to 5'. Once laid down, polymerase seals it together. Once that's done, the ligase will come and connect the DNA back together, combined with the new ones, and now you have 2 DNA strands instead of just 1. For the extraction of DNA in the strawberry experiment, the soaps or salt used in this is used to break open the cellular membrane to release the DNA, while the ethanol is used to clump the DNA together and bring it to the surface.
Polymerase chain reaction, or PCR. The DNA found at the crime scene has 40 base pairs. PCR is a sequence to make many copies of DNA using only a small amount of DNA. The DNA electrophoresis DNA is negatively charged, so they will always attract to a positive. The table shows that the crime scene blood had 40 base pairs in its DNA. Person 1 also has 40 base pairs, while person 2 has 10 base pairs.